Thanks, Jeff. Just a quick confirmation that you can hear me and that you see my slide full screen.
Both. Great for both.
Everything's all set? Okay, great. Thank you.
Sure.
All right. Thanks for the introduction. I'm glad to be here with everybody. And, Jeff makes everything sound great with growing soybeans. And then in comes the plant pathologist who's here to discourage you. No, not discourage you, but, share with you all the problems that you could run into. Sorry, my phone. Oh, all right. Okay, so we're talking about soybean diseases today, and I want to go ahead and start with our foliar diseases before we move into this soil-borne diseases. Just a quick disclaimer, I might be mentioning some products. None of it is supposed to be promotional or exclusionary, or I'm not endorsing anything. It's just these are coming right out of the Cornell guide. And so let's go ahead and get started with downy mildew. This is a foliar disease that probably most of you are quite familiar with. It tends to show up just about every year as long as it's not super hot and dry out. And it's problematic not only because it causes cosmetic issues on the leaves that can affect photosynthesis, but it also, under severe infection, can cause seed quality issues, which is what you see in the image on the lower right there. This definitely favored by high humidity. We see it typically in the upper canopy first on the younger leaves before it moves down. And then the problem with it is that it can overwinter in the field on residues like shattered seeds, and then that's where the inoculum is going to be ready for the next year to go ahead and infect your next crops nearby. The issue with downy mildew is that it's not a true fungus. So a lot of our foliar fungicides don't even work against this. This is considered an oomycete, also known as a water mold. So the only fungicide registered for this in New York is Approach Prima, and it has limited efficacy. But this typically, we don't have enough pressure from this disease to need to warrant any actual applications of any fungicides for management anyways. They're just, certain cultivars have varying levels of resistance, and crop rotations will help break that disease cycle as it's overwintering in your field. The next foliar disease we'll talk about is frog eye leaf spot. This is one that's typically considered more of a disease of the Southeast, but it's been making its way north, and obviously out in the Midwest, it's become more problematic. Frog eye leaf spot is, I think, another one that's pretty easy to identify. It has these very distinctive lesions with the dark red borders, and then in the center you can see sporulation if you look closely enough, if it's humid enough. And this is one that is favored by high humidity and moderate temperatures, and another fungus that will overwinter on residues and provide inoculum for following years. And as this disease increases and spreads throughout New York State, the local inoculum and regional inoculum is also increasing, making it more prevalent year after year. Another issue with frog eye is that it, like many of our pathogens, it has developed races, and they are starting to overcome like the genetic resistance that's within soybeans. So soybeans you'll see in the seed catalogs will come with frog eye leaf spot resistance ratings, but those might be for, say, race 2 of the pathogen. And meanwhile, if you get a breakthrough, it might be because you have a race other than race 2 in your field. And another real concern with frog eye leaf spot is that not only is it overcoming the genetic resistance of the soybean varieties, but the pathogen itself is also starting to become more tolerant or resistant to the strobilurin class of fungicides. And this map here is showing the states where they have confirmed that resistance thus far. We have not yet seen that here in New York State, but I would imagine it would be only a matter of time before those isolates make their way up here, considering how much we're seeing frog eye expand in the state. So when we want to manage frog eye, we want to do crop rotations And maybe if you were still using tillage, that would be a good tool to bury some of those residues to minimize the inoculum. Resistant varieties where available, and fungicides when necessary. But of course, an integrated pest management approach using all of those would be best to help limit those resistance issues. And listed here are just some of the fungicides that are labeled in New York State with good to very good ratings against this particular disease. Another one of our most common fungal diseases, foliar disease, Septoria brown spot. Some people call it Septoria spot, some people call it brown spot, same thing. It's pretty much everywhere. It's favored by warm, humid conditions. Just about every year you see it somewhere. These lesions are just kind of irregular. They cause some yellow chlorosis. Sometimes it's right around the lesion. Sometimes it turns the whole leaf. Um, this pathogen starts typically lower in the canopy and then makes its way upward. And some of the things that are a little confusing about this one is that it's very similar to, say, bacterial blight or bacterial pustule. So sometimes they can be a little bit difficult to distinguish, and getting a correct diagnosis, of course, is going to be key for management, because you don't want to be mistaking this for a bacterial disease and then try and spray a fungicide on it. So the issues with this one is that if it's severe enough, it can cause premature defoliation and up to about a 15% yield loss. But around here, we rarely see it that severe. And once it does start taking off, it's typically towards the end of the season and isn't going to be having, that huge of an impact on your yields. So I would say you're probably not going to get a return on investment in most years spraying a fungicide to control brown spot. So crop rotation and just fungicides when necessary. And the products listed here are those from the Cornell Guide that have very good to excellent ratings. And speaking of those bacterial diseases, we have both bacterial blight and bacterial pustule. And these two are extremely difficult to tell apart, even for pathologists. And you can see how much they look like Septoria brown spot. So again, just sort of irregular reddish brown lesions with some yellowish halos. So the way, one of the easier ways to sometimes tell these apart is that under the right conditions, pustule gets its name because it'll have a raised bump on the underside of the lesions, on the other side of the leaves, but that's not always obvious. But also they tend to thrive in two different temperature regimes. So bacterial blight you tend to see earlier in the season when temperatures are cooler, and it's limited at about 75 degrees. And then later in the season when temperatures start climbing higher, that's when you're more likely to see bacterial pustule because it likes it a little bit hotter. And another sometimes easier way to distinguish this from Septoria brown spot is these bacterial diseases, both of them tend to prefer younger, more tender succulent leaves. So you'll see them in the tops of the canopies before you'll see them down lower. And I would say that you're very unlikely to ever need to make any management decisions. However, if you're growing a variety and you think that this has just exploded in your field, you might want to just switch to a different variety because there is some variability with resistance. Another fairly common fungal disease that we see sporadically around the state is Cercospora leaf blight. This is it looks very similar to what we would call sun scald, and it can be a little bit problematic. It can cause early defoliation, which can affect yields, but really the biggest concern with this is that purple seed stain that you see down in the right-hand side. And this purple discoloration is actually caused by the fungus. It's a secondary metabolite toxin that it produces that's translocated throughout the plant, and that's what's causing this purpling of both the leaves and the seeds. Um, Management can be a little bit tricky. Fungicides are not recommended. There have been a lot of trials on this. They don't work particularly well, but they do offer some protection. So if somebody's growing, say, edible soybeans where the purple seed stain is really a concern, or maybe a seed production field, fungicides might be warranted. But the best, management is really to just kind of, if you grow a variety that seems susceptible, maybe move away from that variety. So moving on to soil-borne diseases, we're going to go through a few of these. We're going to start with white mold. Jeff told you about this, and anybody who's been growing soybeans for any period of time typically ends up having an issue with this at one point or another, especially in years that are particularly conducive. This is definitely the highest concern for yield loss in New York State. It's very widespread and it's unfortunately undermanaged. And the reasons for that is that the fungus produces these little structures called sclerotia, and they're very long lived in the soil. We've seen research of up to 20 years or more. They recover viable sclerotia in fields that have not had a susceptible host in them. This disease is favored in those years where we have kind of cooler, below normal temperature and high either humidity or precipitation. You tend to see this pop up at canopy closure during flowering because that's when you're going to have the highest humidity within the canopy. And another problem with this particular pathogen is that it has an extremely wide host range. Most dicots, including a lot of the weeds that you have in your field, so making crop rotations a challenge for, to rely on those alone for management. So if you do want to look at crop rotation as a management tool, minimum of 3 to 5 years really to start to reduce the inoculum. Even a single year out of it is helpful, but the 3 years is really kind of the target. And we want to start looking at planting varieties that are listed at least tolerant to white mold, but a lot of these aren't widely available, but the seed companies are getting better with this. It's just a really challenging disease to develop varieties with resistance to it because of the multigenic resistance factor to it. Canopy management, if you're planting on narrow rows, you're more likely to get disease than in the wider rows. I'll show you a slide on that in a second. There's another management tool is biocontrol. There's a product called Contans, and this is a parasite. It's another fungus that parasitizes the sclerotia and degrades them, but it's rather pricey, and it does require both a financial and a time investment because it takes multiple years of applications, but it has been proven to definitely decrease the inoculum in the field. And some growers are, they swear by it. So, I would say if this is an issue for you, go ahead and give it a try in combination with all the others. And then, of course, fungicides are probably our best tool right now. And some of them do require multiple applications according to the labels. Some of them have single applications, but timing of those applications is critical and tricky. And it's probably one of the biggest questions I get all the time. Do I put it on at R1 or do I put it on at R3? And, unfortunately, the answer is tricky because it depends on the year, it depends on the weather forecast. And you have to take all those things into consideration. So Jeff had mentioned nozzles, whether you're doing it for herbicides or fungicides. When we're trying to get penetration into the canopy, we want to have the right kind of nozzles to get down in there. So we don't always want to be using those same herbicide nozzles when we're switching over to spraying fungicides. You're just going to get better application coverage when you use the correct nozzle. And this on the left-hand side, this is a bit of a busy slide. This is from North Dakota State University. There's a lot of information going on in here, but basically these are fungicide trials with a few different products. And the top graph is looking at the disease incidence of white mold. And then the bottom figure there is looking at the effects on yield on those. And so to simplify things, and this is based on row spacing. So we see 7-inch, 14-inch, 21, and 28-inch. And to simplify this for you, we want to say pink is good, okay? So if we're looking at disease incidence, we can see that there's most pink in the Endura at 8 ounce across the bottom. And then also we're seeing less disease incidence in the 21 and the 28 inch row spacing. So, and then if that does translate to yield as well. We see Endura worked a little bit better than Approach and Proline here. And the 21-inch row was especially good here for this one. So again, once you get this disease, we want to look at an integrated approach with row spacing, resistant varieties, possibly biocontrol, and fungicides. Fusarium wilt. This is a disease that has been popping up all over the state in recent years, especially in the very dry years. This is one that will show up, and we did confirmed it for the first time in 2012, though that doesn't mean it wasn't here before that. This one, it's like the infection occurs in a cool, wet spring, and then the symptoms show up if the weather then turns hot and dry. According to its name, you'll start seeing wilting. And this is another pathogen. It's a fusarium pathogen, and it's long lived in the soil. So crop rotations are challenging, but still somewhat effective. And you'll know when you do a drive-by on this, you look over at the field and you just see these drooping, wilting plants, and you'll think, what the heck is going on out there? And if you cut open the stems, especially at the lower stems, what you see in the photo there is it's not necessarily the pith that you're paying attention to that's discolored, but it's the outer vascular tissue you'll see has turned brown. This disease is sometimes, especially in the field, easily confused with Phytophthora, Phytophthora root rot, which you sometimes see because, as you can see in the field picture there, it kind of looks like what Phytophthora will do to a plant. So management strategies for Fusarium wilt include reducing compaction, and if you know you have a field with a history of this, it could be problematic any year because it's so long-lived, so you want to maybe consider delaying your planting until your soil temperatures are favorable for emergence to help it outgrow that early stage of infection. Crop rotation will be helpful. Foliar fungicides are not effective against this because this is a soil-borne disease. Next, we're going to move into 3 diseases that I think are really probably some of the most confusing ones to a lot of our growers and consultants here in New York State. And the reason is because they can look very, very similar, and until you really hone in your diagnostic skills, you could easily confuse one for another and make a bad recommendation for management. We got sudden death syndrome, northern stem canker, and brown stem rot. And what you see up in that top center photo is typical intervenal chlorosis, intervenal necrosis. And, it's just kind of like, this is what all these diseases can do to the leaves. This is a bit of an extreme case here, but just so you know that they can all do this, but they have different other symptoms, including what they do to the roots versus the vascular system versus lesions on the externals of the stem. And then the management, of course, for each of these diseases is going to be different. So let's start with one of these, northern stem canker. So you see here in the photo that the leaves there at the top, that's that typical intervenal chlorosis, which someone might say, hey, that's sudden death syndrome, when in fact it is not. This is northern stem canker. This was first confirmed in New York back in 2014. And since we first confirmed it, we have been seeing it just expanding a lot across the state, out in Western New York and Central New York. Here in Tompkins County, we seem to be a hot spot for it, and some of the fields around here just get hammered with it. So this is another one of those diseases where it's in the soil, it's going to overwinter in the residues, and it's going to be able to stay in the soil for a while. Infection occurs early in the season, but you don't see the symptoms until later. And when it is severe, you can have up to 50% yield losses. So this is one that's almost rivaling white mold, as far as a concern that we want to pay attention to. And it's important to know that I'm emphasizing northern stem canker because there are 2 different variants of this disease, northern and southern stem canker. They are 2 very different diseases caused by 2 distinct pathogens. separated typically by geography, although we did confirm southern stem canker up here in New York as well, but by far northern stem canker is the biggest problem. And it's also important to know that when you're looking at those seed catalogs, at least for now, hopefully they get this addressed in the future from some of the major seed companies, is you'll just see it listed as stem canker in the resistance ratings. And that, I would say, up till now and moving forward, is always referring to southern stem canker. It has nothing to do with northern stem canker. As far as I know, none of the companies have released varieties that have resistance to northern. So this northern stem canker is something that if you were just doing a drive-by scouting and you saw these dead plants out there in your field, you might think it's something else. You might think it's sudden death syndrome, but oftentimes I get called, people think it's white mold, and then they get out there and they say, where's the fuzzy white stuff? Because you'll just see these patches of dead plants, which is what you see with white mold. So that's why you have to look closely, make sure you're getting the right diagnosis. Management for this is crop rotations and maybe tillage if you're using tillage to bury the residues. And hopefully in the future, we'll have some resistant varieties because fungicides are not recommended, at least none of the ones that have been evaluated in trials across the country. They don't have very good efficacy. They call it fair efficacy and or fair to poor. And so they're not recommending fungicides for this. So right now it's a bit of a challenge and it's growing and we don't have great tools to deal with it. Somewhat related to northern stem canker is part of the disease complex is called pod stem blight. And that causes Phomopsis seed decay, which is what you're seeing in that picture there. These moldy, yucky, cracked seeds that are white, they're unmarketable, they won't germinate, they're terrible. This is very widespread. You tend to see it late in the season, those black specks along the stem. That's actually the fungal fruiting bodies. In most years, you'll see it at some low level at the end of the season as the plants are maturing and senescing, but it's rarely an economic issue for us here because it does come in so late. Typically, those plants have already done what they need to do, and as long as you get them harvested in a timely manner, you're not likely to see too much damage with this Phomopsis seed decay. So another one of those three diseases that's easily confused by those foliar symptoms is brown stem rot. You can see this nice photo here taken by Mike Stanyard. Again, you might think, hey, that's sudden death syndrome, or no, that's northern stem canker. But nope, this one's actually brown stem rot, looking just like the others above ground. This is another typical disease favored by cool, wet spring, followed by a hot, dry summer, because as you can imagine, it's clogging up that vascular tissue. So once the water shuts off and things dry out, those plants can't move their water nutrients up and down for proper respiration. And with this one, you won't see any of the external stem lesions that you would see with stem canker. But what you will see is if you split these stems open, you'll see this chocolate brown typical infection in the pith of the plants, and that's very indicative of brown stem rot. Fortunately, this disease is very well managed with genetic resistance. The majority of varieties available, even just in our maturity group ranges, should have pretty good brown stem rot resistance. So if this is something in your field that you have a problem with, go ahead and go talk to your seedsman and make sure you select a variety that has ratings for good resistance to this disease, and you should be all set. Because if this is a soil-borne disease, foliar fungicides are not going to work because the infection happened through the roots early in the season, and doing a revenge spray on these is going to do nothing for you but hurt your pockets. And then that brings us to sudden death syndrome. This one's a little bit trickier. This was first confirmed in 2012. Again, this is a disease that's more common in the Midwest or the Southeast. So when it popped up here, it was a little bit of a surprise. And then, of course, once it's here, it just started making its way around and it's expanding. And now it's almost considered common here in the state. This is, as with the others, favored by a cool, wet spring, followed by a hot, dry summer because it's affecting the vascular tissue in the plants. For this one, the infection occurs very early, sometimes almost as early as germination, and then you don't see those the symptoms until the reproductive stages. This one has, in my opinion, some of the most striking intervenal chlorosis symptoms. And what this one is doing is actually the fungus is inside the plant in the vascular tissue, and it's releasing a toxin, and that's what's causing that yellow flashing. And so to diagnose this one, you want to look on the outsides of the stems. You won't see any lesions. So northern stem canker is the only one, as you could tell by its name, that's going to have external lesions visible. So sudden death syndrome does not have that. And then when you split the stems, you're not going to see that chocolate brown that would be indicative of brown stem rot. What you're going to see is browning of the external vascular tissue, which is typical of many fusarium. So, and this one is also often associated with soybean cyst nematode. They make each other worse in the field. Another symptom you may see is in this upper right corner, only if you're lucky. If you pull up the roots, you might see the fungus, which is bright blue, and it may be sporulating on the roots. I've seen this in so many photos and books, and I've seen a ton of sudden death syndrome infected plants, and this was the first time ever and only that I dug up a plant and actually saw this blue sporulation that everyone describes. So don't rely on that, but when you do see it, it's pretty striking. For managing, of course, we need to do correct identification. Foliar fungicides aren't going to do anything for you with this one. Reducing compaction when possible. Crop rotations are good. Resistant varieties are available, but they're probably limited in early maturing varieties because, they might be working on that, but they haven't typically bred for resistance for our region because it wasn't typically an issue in our region. But seed treatments, there are a few seed treatments now on the market that are available and showing some really great promise and may be worth the investment if you're starting to see more and more sudden death syndrome around. And then, of course, once you know you have an issue with soybean cyst nematode, that's something you also want to start managing. So I want to look at some data here on these seed treatments for sudden death syndrome because we're starting to get more and more questions about these. So this is from Michigan State University. They did replicated trials across the state. I think this is 8 different locations. And here for these examples, they had very low disease pressure from sudden death syndrome at all the sites. And with that being the case, with low disease pressure, only one of those 8 sites actually showed any improvement on disease reduction or yield compared to the non-treated control. So overall, in these situations in low disease pressure, the seed treatment did not pay off. If we start looking at another trial where this was multiple years, the same trial is replicated over multiple years, 3 years. Basically what this is showing is that over those 3 years, these growers had about a 50% chance of getting a return on investment for investing in that Ilevo seed treatment when they only had low disease pressure. So driving that message home again. And here is from University of Illinois, looking at sudden death syndrome. And this was with moderate disease pressure, and it did significantly reduce the disease and increase the yield because there was obvious moderate disease pressure. This one being another one from University of Illinois, and this one was low disease pressure, and it did reduce the disease but didn't impact any yield, so probably no return on investment there. And then there's a new product also. So Ilevo hit the scene first, and then the new product came out called Saltro, which is going to provide similar, if not better, protection against sudden death syndrome as that new Ilevo product. It's definitely better than the typical seed treatment packages that you're seeing. But again, this will probably be under, at least moderate disease pressure. And here's another one from Kansas where they're comparing Saltro and Olivo. Both of them were better than the control as far as reducing disease and increasing yield. Saltro did a little bit better than Ilevo in this trial as well. So now that we've gone over that, the next question might be, do we need those seed treatments for sudden death syndrome here in New York State? And the answer is, well, maybe. It's going to be on a field by field situation. As we just saw, they're only going to be cost effective under moderate to high disease pressure. And of course, they'll be even more helpful once you know that you have also soybean cyst nematode in addition to your sudden death syndrome. And are we headed that way? It kind of seems that way. Both of those problems are becoming more and more prevalent. So we need to start looking to that. But so for now, let's look at an IPM strategy where we try to select and plant resistant varieties, or if we can't find resistance, at least early maturing varieties. Manage your compaction and your drainage, and keep an eye out for soybean cyst nematode, which is what I would like to talk about with you guys next, is soybean cyst nematode. This is definitely considered the #1 pest in the US, where it causes over 109 million bushels lost per year on average each year. And then also even globally, this is in many years considered the #1 pest causing losses. And what this map is illustrating, this is from 2017. They've been tracking the counties that have shown positive results for soybean cyst nematode. And what you see is it's got really great, or not great, but a lot of distribution across the Midwest, North Central region, and down in the Southeast. And you'll see even New York got put on the map in 2017. But if we look at this a little bit more closely now, so we first started testing for soybean cyst nematode in 2013 here in New York State. These maps here, first of all, to give you some clarification, green, counties colored green mean in that year, the counties were tested but did not have a positive result. And then any counties that are red were positive, and gray just means they hadn't been tested. So like I said, we started testing in 2013. It wasn't until 2016 that the Cornell lab was able to identify one single field that happened to be in Cayuga County that was positive. And we continued a little bit of testing along the way, small numbers of fields per year. And then in 2019, when I started with the New York State IPM program, we decided to work with New York State Department of Ag and Markets and do some more systematic testing and sampling specific to soybean cyst nematode. We started working with a lab that really specializes in this. And out of 25 samples, we identified another 6 counties. It was more than 6 fields, but 6 counties that were positive for SCN. And that, of course, was big news because for many years we thought it was just in this one county, or at least that's the only place we confirmed it. If we were probably assuming it was more widespread. And that spurred us to do some further testing in 2020. We got support from New York Corn and Soybean Growers Association and New York State Department of Ag and Markets again. We tested 125 fields, and I think we got up to, was it 26 counties here that are positive. So this can't be ignored anymore. Nobody can say that soybean cyst nematode is not a problem in New York State. We know that it's here. But the good news is that most of those fields that tested positive, they were at low population densities. So that's good because it's, we want to keep them low, but there definitely were a handful that were in the moderate range. And there was one field that was very high, and that was rated at 40,000 eggs, and that's per cup of soil. So you see that the range is down below, that's less than 500, 500 to 10,000, and greater than 10,000. These are per cup of soil. So if you can imagine finding 40,000 eggs in a cup of soil, that's pretty bad because most of the fields that we found that were positive had about 150 to about 350 eggs per cup of soil. So why do we care? Well, we already discussed that soybean cyst nematode is the number one problem. And it's important to know that eradication, once it's here, is not an option. You're never going to get rid of it. It's very long lived and persistent in the soil, and that provides a lot of management issues. And if you look at this image here, this is an aerial image of a typical field in Iowa that is infested with soybean cyst nematode. And that's it looks pretty severe and awful, but unfortunately, this is almost commonplace in areas that have been dealing with this pest for decades. So we're poised in a very good position right now to try and stay ahead of this pest because it's a heck of a lot easier and a lot less expensive to maintain your populations at low or even moderate levels than it is to be reactive and realize that you've somehow let them get to high and then trying to reverse that trend. So these little nematodes are quite prolific. I love this infographic because it really, I think, drives home the message of how quickly this thing can become problematic in a field. So one single cyst can contain about 200 or 250 eggs. So if we start at the left-hand side of this image, we have um, say one cyst on one plant, right? And that's going to hatch or burst with eggs. They're going to hatch into juveniles, males and females, and it's just going to go on and on. You can follow the math as you go. This is showing for 3 generations, and here in New York State, we can have 3 generations. So this is very typical of what we would see here. We can go from 200 eggs to over 39,000 eggs on that one plant in one season. And that's pretty intense and not a situation that we want to see happening. So we want to start managing for this. Soybean cyst nematode is definitely a yield robber. It can be a silent yield robber. We say that because you can be losing 10 to 30% of your yield without any above ground symptoms. You don't even know that it's happening. You're thinking it's something else. Why are my beans yielding low? You know, is it fertility or something else? You won't even see it. You won't even notice it. So how do you know if you have soybean cyst nematode? Well, testing is critical. The above ground symptoms can oftentimes be very vague or entirely absent. So if you see this field here, if you were doing a drive-by on that, soybean cyst nematode might not be the first thing that pops into your head, right? You might think, oh, that's a nitrogen problem, or you know, some other nutrient deficiency, or it might be a wet spot in the field, but that's actually soybean cyst nematode. And so the fields when you don't know that you have it yet and you're just looking to confirm it, you want to focus your sampling on fields and areas of fields that are most likely to be infested, because it can be really hard to find it and confirm it in when the populations are really low. So if you have fields or sections of fields that are consistently low yielding that can't be explained by things like fertility or moisture issues, that would be a good thing. Fields that have a history of sudden death syndrome, we talked a lot about sudden death syndrome. There's a very tight correlation between that disease and this pest. So those are good ones to concentrate on. And then focus on areas of the field that are compacted or prone to flooding. The nematode seems to like that. So the image on the right here is just emphasizing those areas of the field where you might want to focus your sampling, where you'd be most likely to find it first. But then even so, you might test a field and get a negative result, but that doesn't necessarily mean that you don't have it. It just means you didn't find it. And this image on the left here is showing you how easy it can be to miss it. And, you know, so there's a little clump of cysts there that's supposed to be representing a soil probe going into the soil. If you miss that clump of cysts by 1 centimeter, you could have results that say 0, meaning, oh, I don't have a SCN in my field, or you get those cysts and you get an egg count of 1,500. So it's really challenging sometimes to get, you know, your first confirmation, but you just got to be vigilant. And to help explain that further, soybean cyst nematode is very variable in a field, like most of our soil-borne diseases, like the image I showed you from that Iowa field where you saw the patches of dead plants in the field. It's just like this image here on the right. It's like a heat map of the intensity based on some pretty specific grid sampling in that field for soybean cyst nematode. And you can see where if you only did one section of the field, you might get super high numbers. If you only did another section, you might get nothing at all. In addition to soil sampling, you can also do visual checks during the season, during the growing season. To do this, you really have to know what you're looking for. And I'd say you want to dig the plants up. You definitely don't want to pull them. Those cysts can be dislodged rather easily. And you want to gently remove the soil and look closely. And better yet, if you even have a little bucket of water to bring with you, you might want to dip it and get those roots as clean as these ones are here so you can see. They definitely look more like little grains of sand and are much tinier. You see them here as compared to nodules. And you see the little white bits over here. And because of the reproductive cycles here in New York, the best time for you to be digging roots and inspecting them for this will be mid-July or early September. But otherwise, you want to do soil sampling, and I would recommend doing fall soil sampling either at or just before or just after harvest. It's best to sample within the rows, and you want to stay in the top 8 inches with your soil samples. So once you have confirmed that you do have SCN in your field, you don't have to do that targeted sampling anymore where you focus on the bad spots. And then you can transfer over to more random pattern sampling. If you have a nice big squarish field, you break it into 10 to 20 acre blocks and do kind of very systematic random sampling. But most of our fields look more like the lower image with topography and such. So just kind of do a squiggly through broken into areas no larger than 20 acres. And once you confirm that you have it, you want to keep track of your numbers and you want to keep monitoring those fields every 3 years and see how your eggs are changing, whether your counts are going up or going down. And you want to record and track those egg counts. I know nobody wants one more thing to keep track of, but it's going to be very helpful for you to keep an eye on those numbers because that's going to be how you, that's going to determine your best management recommendations. I get asked all the time, where do I send my samples? There are a ton of labs out there, both public and private, that offer testing for soil testing for this nematode. I highly recommend that you work with a lab that kind of focuses on this and specializes expertise in nematode testing. We work with a lab called SCN Diagnostics. I think the name says it all. They're at University of Missouri. They do accept out-of-state samples. They are one of the cheapest ones at only $25 per sample. They're extremely fast. Like you send something on a Friday, you've got results on Monday. They are very responsive. They'll give you the egg counts that you need. Be careful. Some labs will just give you a positive or negative. You really need egg counts. And also this lab, if you're ever curious in the future, they can provide the race testing because this nematode does develop different races, but I don't think that that's very critical for us to know what the races are. So what do you do once you've got a positive sample? Well, we already went over testing, so keep testing and keep track of your numbers. Next, I want to cover crop rotation. This is a really important tool and a really great one for us here in New York State because we already, many of us, are doing some great crop rotations, whether it's small grains or corn or hay or something like that. So rotation in non-host crops. In this example here, they're saying that in the first year rotation to corn, you can see up to a 50% decrease in the nematode population. Now, of course, the decrease in the nematode population is going to depend on the intensity of the nematode population to begin with. But what they're also finding is that subsequent years of rotation out of soybeans, they still have an effect on reducing the nematode populations, but it's never as dramatic as that very first year. So crop rotations are a really good tool. But SCN is very long lived in the soil, and that causes some problems. And so if we look at the right-hand side of the screen here, these egg hatching behaviors, this is part of the reason it's so long-lived, because it has 3 different strategies for egg hatching. Some of it, they hatch, they'll just hatch consistently, whether there's a host or not a host present. There's some that only will hatch in the presence of soybean root exudates. And then there's some that just go dormant for an undisclosed number of years and will just pop up, who knows how many decades later. So if we look at the examples on the left-hand side, this is when you start realizing how alarming this could be. The first one there was a field that they cropped it to fescue, which is a non-host grass crop. And after 40 years of not having soybeans in that field, they found viable SCN eggs. And so 40 years, that's a pretty long time, which is why we say eradication is never an option. And then in the next example there, we see this field that was known to be SCN infested. They kept it fallow and weed free for 8 years, planted beans in on year 9 and then again in year 10, and they saw yield losses and damage 10 years later in the soybeans. So crop rotation is great, but it's not going to be perfect. And another reason it's not perfect is because soybean definitely isn't the only host for this nematode. And dry beans, I heard also Jeff said that you guys are growing some snap beans out there. So any edible bean crop will be susceptible to SCN. Some of other things that, we're getting into, say, cover crops. We have clovers and vetches. Some of those are hosts and will increase those SCN populations in your field. There are some non-host crops. Alfalfa, fortunately, is a non-host crop. Some of the clovers are non-host, but also, look at corns, any grasses, small grains, things like that. But one of the other things to keep in mind, something that will keep these critters reproducing and happy in your fields, are the weeds. If we look at this list of weed hosts, we pretty much have all those here. So again, just contributing to the inability to eradicate this pest. So let's look at crop rotation. Let's use it as a tool, but let's understand that it's not a standalone solution. Then let's look at the resistant varieties. The good news is that so many soybean varieties that are available already have labels as SCN resistance, and the seed companies aren't charging you anymore to buy a resistant variety versus buying a susceptible variety. So it doesn't even cost you anything. So they're widely available, they're really cheap. That sounds perfect, right? And these resistant varieties, they have a double whammy effect on this as far as being positive for you. They both can increase yield and they're reducing the reproduction of the soybean cyst nematode in your field. Because the way the resistance works, the biochemical pathway, is that it stops or minimizes the nematode's ability to reproduce. And if it's considered a resistant variety, it should be holding reproduction at less than 10% of what it could be. So that sounds so perfect, right? Why don't we just plant resistant varieties and just call it a day? Well, it's because, as with anything that too much of anything is, can be problematic, just like the issues we've had with herbicide resistance over the years and, some corn insect pests overcoming some of our BT traits. The nematode has started overcoming those resistant traits. And the reason is because most of the varieties that were released that are called SCN resistant all came from the exact same source of resistance. It's known as PI88788. And this map here is showing the percentage of nematode populations that are overcoming that resistance in those states. So if we look at that in another way, this is all the varieties that have been released over the past 20 years or so that are claiming SCN resistance. And it's great because there's a lot of them that are out there. So the gray portion of each of those bars represents the percentage of those varieties released that all came from that single background, PI88788. They have the exact same resistance. And then red is coming from some other. So you can see what a small percentage of all those actually has some other different source of resistance. So that's problematic with the resistance parts and why it's not a silver bullet. And so then let's move on and look at these nematicidal and nematode protectant seed treatments, because there's a ton of products available. Some of them are biological, some of them are chemical, some of them are plant derived, some of them are very specific to SCN, some of them are very broad in general against all nematodes. And so, you know, we have a lot of variety out there. This is, to the best of my knowledge, the latest list of products that is available. We see a lot of the active ingredients here, anything from biologicals like bacillus, or which a lot of us are familiar with, down to some proteins and also some chemicals. We have a couple here, Ilevo and Saltro. You'll recognize those names because we went over seed treatment trials for those for their efficacy against sudden death syndrome. These are actually SDHI fungicides, fluopyram and flumetafilone. I'm struggling right now. Anyways, those two chemistries, SDHI fungicides, and but they have shown some nematode suppression as well, so they're being counted as these nematicidal seed treatments also. But it's great to know that we have all of these tools, but they do tend to show some variable results. Just like we saw with the sudden death syndrome, you know, it all depends on the pest or disease pressure. The lower the SCN population pressure is, the less likely you are to have a return on investment for these things. And you know, the scientists are saying, this is a great quote from one of the SCN gurus, is treatments might reduce the SCN reproduction, they might increase yields, they might do both, or they might not do either of them, you know? So it just depends on the situation. They can vary among soil types, locations, growing seasons. So it's a little bit tricky to make strong recommendations with these, but they are great tools, especially in areas that have high levels of SCN. But they are good tools, and they can help us slow the breakdown of that SCN resistance in our in our soybean varieties. So we got to pair these things together. Let's look at a little bit more research on these. I put in red type here the names of the products in case you didn't remember the biological active ingredients. So if we look at this, we've got Aveo, Votivo, Clariva, and Ilevo here. And this is on dry beans here. So, you know, but these are, it's the same thing, whether it's soybeans, dry beans, or snap beans, the disease or the pest is the same. And we see in this instance, none of these treatments had any benefit over the non-treated control. And this was a field that had natural levels of infestation. If we look at another trial here from Michigan State, this is looking at that Ilevo. This is the same trials that we looked at with sudden death syndrome, but this is looking at the nematode, and we're looking at SCN reproduction from planting to harvest. And what we see is that this Ilevo seed treatment really had very mixed results among the locations, from no difference at all between start and finish, between the control and the Ilevo seed treatment, to significant reductions to significant increases. So again, this is what we say, where the data is a little bit sloppy, and sometimes it works great and other times it's, you're just not sure that it's doing anything. So we get questions all the time, what about those seed treatments? Should I be using them or not? Well, there can be an inconsistent return on investment, and I would say you probably don't need to be investing in them until you get moderate to high SCN or SDS, or well, in this case, SCN populations in your field. And of course, all this survey work can't be done by one person alone. So I want to acknowledge everybody involved, including all the extension folks like Jeff Miller, funding from New York Corn and Soybean Growers Association, New York State Department of Ag and Markets, also Northern New York Ag Development Program. Thanks to the New York State IPM program for coordinating all of this, and SCN Diagnostics for doing such a great job providing us with all the testing. And with that, I guess if I have a minute, I can take questions, or Jeff can take it from there and pop up the CCA code.
Thank you very much, Jamie, for contributing so much to today's program. I did post the end registration link for those of you that want DEC credits. Please take the time to get into that chat box again, click on the link, and then fill out the necessary information on your way out. One of the questions I have for you, Jamie, is, okay, so I've heard that leaving that sclerotinia of the white mold on the surface, the soil surface, is also a way to try to at least lower the numbers. What can you comment on that?
Sure, that's one of those tricky questions because the pathologists seem to fall into two camps with this one. We have the ones who say no, bury it, and then we have the others who say no, leave it on the top. And I think that there's validity to both of those strategies. So for those who are saying to bury it, it's because if you bury it, it has to be within a certain depth in order to be able to germinate and then send out its little apothecium mushroom and then spread its spores. So if you bury it deep enough, then they can't germinate and they can't infect your crop the next year. But we also know that it can be there for 20 years or more. And so if you ever come through there with tillage again, you're going to bring them right back up to the surface possibly, and then they can germinate. But also while they're buried, they're more susceptible to being broken down by microbes and things like that. Then the other camp is leave them on the surface, which Jeff just said they can be more subjected to breakdown by UV. And that is a true thing and it's a great thing, but also you're leaving them right there on the surface. So even if you're not growing soybeans the next year, if the conditions are right, they're going to possibly germinate and blow into the next field over, across the street, your neighbors, whatever. But I think both of them are decent strategies. And I don't take a side either way. I think that it's going to have to be whatever you feel more strongly about and how you're doing it. Obviously, if you want to invest in that Contans biocontrol product, you're going to want to leave them on the surface because that's when you're going to be able to get them covered with that biocontrol product. So Jeff, do you have a preference, one or the other, or what are you recommending?
No, it makes sense that if you're moving towards or in a no-till program, then you're obviously going to err on leaving it on the surface, right? Mm-hmm. And if you're already doing tillage, then maybe your comments related to using tillage as a mode would be something to consider as well. So I'm glad to get the clarification on the two camps.